Today we're going to look at Module 6. For a brief introduction, we're going to be looking at processes of reproduction and genetics. Genetics is just the science that studies how characteristics are passed from parents to offspring. We know that this passing of traits from parents to offspring requires instructions or a blueprint, and we find these instructions in the genetic storehouse of DNA. So the genetic storehouse of information that governs traits and characteristics is just deoxyribonucleic acid, or one of the more common nucleic acids, DNA. There are determining factors that all work together to determine what kind of person you are. Genetic factors, environmental factors, and spiritual factors. Genetic factors are the most easily studied from a scientific standpoint, and they're just a general guideline of traits determined by a person's DNA. Environmental factors are non-biological factors, and they include things that are in someone's surroundings, like the nature of the person's parents, friends, or their behavioral choices. And also, they include things like exposure to chemicals, mold, extreme weather conditions, and allergies. And spiritual factors are the factors in a person's life that are determined by the quality of his or her relationship with God. And we're going to focus on the genetic factors just because they're the easily or most easy to study. So let's do a quick review. DNA is deoxyribonucleic acid, and simply put, DNA is the genetic blueprint that we read to make a protein. There's this idea of one gene, one protein called the central dogma of molecular biology. That means we use DNA as the instructions, RNA to carry out a lot of the processes, and that all helps us make a protein at the ribosome. But before we delve any deeper into DNA replication, its role in protein synthesis, and eventually cell division, let's review. So, DNA is found in the nucleus. It's a genetic library with tons of genes, which are the blueprint for individual production of protein. It can respond to signals and dictate kinds of amounts of proteins to be synthesized. And proteins are important because they can be both structural and catalysts within a cell. Many cells have one nucleus, but some have two. If a cell has one nucleus, that's uninucleate. If it has two or more, it's multinucleate. Something like a skeletal muscle cell has many nuclei. And any cell without a nucleus is considered anucleate. An example would be red blood cells. There's three general regions to the nucleus. We have the nuclear pores within the nuclear envelope, and then finally the nucleoli, which is the dark staining region where ribosomal RNA is made. Looking at nucleic acids, we can kind of take a step back. Nucleic acids are a biological polymer that have monomers called nucleotides. All nucleotides are composed of a nitrogen-containing base, a pentose or 5-carbon sugar, and a phosphate group. The two major classes of nucleic acids are DNA and ribonucleic acid or RNA. The nucleic acids have these 5 nitrogen bases. However, uracil is only found in RNA, not DNA. Uracil actually replaces thymine in RNA. The bases are adenine, guanine, cytosine, thymine, and uracil. DNA is a double helix. We find that on the outside of this double helix, we have a sugar phosphate backbone with inner nitrogenous bases. DNA holds the genetic blueprint for the synthesis of all proteins, and its nucleotides are made up of the sugar deoxyribose, phosphate groups, and one of the following 4 nitrogenous bases: guanine, cytosine, thymine, and adenine. Now, if we look at the bonding of nitrogen bases, especially in DNA, we can see that they follow base pairing rules and Chargaff's rules. So base pairing rules are just that A always pairs with T in DNA, and G always pairs with C in DNA. So that's adenine always pairs with thymine, and guanine always pairs with cytosine. Because of these base pairing rules, if we're given information about how much adenine or how much cytosine is within DNA, we can figure out how much of the other bases is within DNA. For example, if there is 25% of a DNA molecule made up of adenine bases, then I know automatically that 25% of that DNA molecule also is made up of thymine bases. And if it's 100% for the amount of bases that you would find in a DNA molecule, 25% is also guanine, and 25% is also cytosine. So thereby giving just some basic information, I can kind of extrapolate using basic math, how much of each base is located in a single DNA molecule. So here's the basic structure of DNA. It's a double helix sugar phosphate backbone with bases in the middle. The bases are held together by hydrogen bonds. And again, adenine always pairs with thymine, and guanine always pairs with cytosine. Whereas ribonucleic acid is single-stranded. It's a single-stranded molecule that's found in both the nucleus and the cytoplasm of a cell, and therefore it's able to link DNA to protein synthesis, because DNA is just stuck in the nucleus and it can't leave. So we'll use RNA as an intermediary. It contains a ribose sugar instead of deoxyribose. It uses uracil instead of thymine. And we have 3 different varieties of RNA that help us carry out the process of protein synthesis. Those would be messenger RNA, called mRNA, transfer RNA, called tRNA, and ribosomal RNA, called rRNA. You can also see here listed the nucleic acid bases of RNA: GCUNA, or guanine, cytosine, uracil, and adenine. All right, here's an animation that you can watch. Now, I'm not going to play it just right now, but I'll make sure that this lecture PowerPoint is posted so you can watch this at a later time. Let's look at a brief history of the discovery of DNA next. Well, in 1869, Swiss chemist Jonathan Friedrich Miescher discovered the substance of DNA, but he called it nuclein because it came from the nucleus of cells. Then in 1881, German biochemist Albrecht Kossel discovered the composition of DNA, so he actually identified the 4 different types of nucleotides, the ones that contained guanine versus cytosine and adenine versus thymine. He actually received a Nobel Prize for his work in 1910. In 1950, Erwin Chargaff, who we named the Chargaff rules after, discovered that adenine and thymine, as well as cytosine and guanine, were always found in a 1-to-1 ratio in DNA. This suggested the base pairing rules, that adenine always pairs with thymine and that cytosine always pairs with guanine. So Chargaff's discovery is known as the rule of base pairing, or Chargaff's rule, for that reason. In 1953, James Watson and Francis Crick built a model of DNA, and this model of DNA was revolutionary because we really didn't know what DNA looked like. Were the bases on the outside? Did we have not only a double-stranded, but were they held together by covalent bonds? We really just didn't know. They used information from Chargaff's discoveries and X-ray pictures of DNA taken by other scientists like Rosalind Franklin and Maurice Wilkins in order to determine the structure of DNA. That it was a twisted double helix with a sugar phosphate backbone on the outside, and all the nitrogenous bases were internal, or on the inside. And that was a tremendous discovery for its time. In the 1950s, Alfred Hershey and Martha Chase did plenty of experiments that were trying to confirm that DNA was indeed the means of heredity, because we really weren't sure until the later 1950s or later in the 1900s whether or not DNA or proteins was what was being inherited by offspring. We were unsure. So that was also a pivotal discovery in DNA and looking at nucleic acids. So DNA was, in the 1950s, confirmed as the means of heredity, what was being passed down to offspring. So let's talk about what's passed down to offspring. It's genes, it's genetic information from DNA. The gene itself, a segment of DNA, is the basic unit of heredity. It's a section of DNA that contains the code to produce a protein or a portion of a protein, and that protein thereby could cause a trait. Now, chromatin is the long strands or clusters of DNA and proteins in the nucleus of a cell. This is the slightly unwound version of DNA that exists primarily in the nucleus of a cell that's not dividing. It's essentially uncondensed DNA and protein. These thread-like strands of DNA with protein and some mixture of RNA are arranged in these fundamental units called nucleosomes, and they're packed and pretty easy to read as long as you have the right enzymes to unwind and read genes or these segments of DNA. When chromatin condenses into bar-like bodies that are visible, as opposed to the uncondensed DNA and protein called chromatin that is not visible, we actually call that condensed version chromosomes, and that occurs when a cell needs to or starts to divide. So genes are the basic unit of heredity. Chromatin is essentially uncondensed DNA and protein, and chromosomes are condensed chromatin or condensed DNA and protein. And we're going to see that we require chromatin in order to have something that can be easily unwound and read to make a protein. And then later on, when we want to pass on this information, we'll condense it in order to move this genetic material around more easily. So here's an image of chromatin. So we can see just the DNA double helix in purple up top, and then it starts to wrap itself around 4 histone proteins, and that creates the nucleosome structure, or the beads on a string structure that we associate with chromatin. Without an electron micrograph or microscope, it's really hard to see chromatin. We couldn't see it with a regular light microscope. But if we supercoil and condense chromatin, we can create a chromosome. A chromosome is condensed chromatin, and it's easier to move around. So when a cell divides, it actually turns its chromatin into chromosomes. So let's next look at DNA replication. DNA replication is considered semi-conservative. We're going to take one DNA strand, or piece of DNA, separate the 2 strands, and then add a new strand to the old strands of DNA. Now, oftentimes in a process of asexual reproduction called mitosis, DNA must be replicated before mitosis or this cell division process takes place. So during the process of DNA replication, 2 strands of the DNA molecule untwist and separate, and then each nucleotide strand then serves as a template for building a new complementary strand. Some enzymes are required for this process to take place. DNA helicase separates the 2 strands of the double helix. It breaks those hydrogen bonds. Then we use DNA polymerase to build a new DNA polymer. But the end result will be that we have 2 new DNA molecules, but we formed part of them from the original. So every new piece of DNA actually has an old strand and a new strand. So it was slightly conserved. So here is what semi-conservative replication is. This is very simplified, but we have the parent strand, and we're going to separate the parent strand. You can see that we have one of each of the strands here in red. And then we were going to add new nucleotides that are complementary to the old strand and create new purple strands for each of the old strands. And that's going to give us 2 new pieces of DNA, each with an old red and a new purple strand. So each new piece of DNA has slightly conserved or saved an old piece of the original parent strand or DNA piece. So DNA replication occurs prior to cell division, and the cell actually has to make a copy of its DNA so that when it splits, each of the daughter cells that is produced gets the right amount of DNA or material for protein synthesis instructions. So remember or keep in mind that DNA is double-stranded, and we have to unwind it and unzip it in order to start to replicate it. The area where the strands separate is called the replication fork, and the area where we are actively replicating DNA is the replication bubble. Each old strand has to act as a template strand for the new strand, and we're going to see that DNA replication creates complementary strands. We're also going to use RNA as a start point, and the RNA is actually laid onto a certain part of the separated DNA strand, and it serves as an RNA primer so that the enzyme DNA polymerase knows where to start producing more nucleotides during DNA replication. So DNA polymerase is going to come in and add new nucleotides to form the new strand on the old template strand of DNA. DNA polymerase synthesizes both new strands at one time, but it synthesizes one new strand in the 5' to 3' direction continuously, and the other strand in pieces, and so that's going to be discontinuously. So we call the 2 strands that are being synthesized at the same time either the leading or the lagging strand. The leading strand of DNA is synthesized continuously, whereas the lagging strand is synthesized in pieces. The pieces are called Okazaki fragments, and they have to eventually be glued together with another enzyme called DNA ligase. The end result, though, is still two identical daughter DNA molecules that were formed from the original, and the new daughter DNA molecules have an old and new strand from that parent strand. And this process, again, is semi-conservative because each new double-stranded DNA is composed of an old and new strand to create the new molecule. So here is just a summary of DNA replication. We can see that we have unwound the two DNA strands. The replication fork is right where they're still being unwound. This area here is the replication bubble, because we can see DNA polymerase actively replicating the DNA. DNA polymerase can only work in one direction, and because the strands of DNA are anti-parallel, it only is able to move in one direction on the leading strand. On the leading strand, DNA polymerase synthesizes a new piece of DNA continuously. On the lagging strand, it'll synthesize little pieces of DNA, and that's eventually glued together with DNA ligase. Another animation that you can access by accessing this PowerPoint on Teams, so I'll make sure that it's posted and you can watch this video. So we've looked at the basics for DNA replication. There's a leading and lagging strand. Leading strands are going to be synthesized continuously. Lagging strands are synthesized discontinuously in fragments called Okazaki fragments and glued back together with DNA ligase. We don't need DNA ligase for the leading strand, and DNA polymerase was what was helping us create the new DNA strand on the old as the template. But don't forget that our focus is protein synthesis. How are we going to use DNA as the blueprint to make a protein? Well, DNA is actually able to direct the order of amino acids in a polypeptide. It's able to do that because a segment of DNA holds the code for one polypeptide, and that segment is referred to as a gene. If you read 3 bases at a time on DNA, you're reading what we consider a DNA triplet, which is 3 sequential DNA nucleotide bases that are from the genetic library. Those bases will be either A, G, T, or C, and each triplet is going to specify coding for an amino acid. And we're actually going to have to convert that code into RNA. And on RNA, we're not going to call the code a triplet, we'll call the base pairs a codon instead. And those codons on RNA actually can pair up with an anticodon on tRNA, and all of that comes together at the ribosome to help us make protein. So let's talk about the role of RNA. We'll have to look at mRNA, rRNA, and tRNA, the 3 types of RNA, because RNA is the go-between molecule that links DNA to proteins. RNA can be in the nucleus and the cytoplasm, and RNA can carry the genetic information or the genetic instructions from DNA to the ribosome where protein synthesis takes place. So RNA is the DNA decoding mechanism and messenger. All RNA is formed in the nucleus, but it differs from DNA in that it's single-stranded, has a ribose sugar, and uses uracil instead of thymine. So it is slightly different. We're also going to find that once we make RNA from DNA, there's pieces we have to take out of this RNA or mRNA sequence. Those are going to be the introns or non-coding segments that must be removed from mRNA before it's processed into a protein. Instead, on genes, exons are the only thing that code for amino acids. So not all RNA is perfect as soon as it's made. So let's focus next on the 3 main types of RNA. Messenger RNA is what is the main intermediary between DNA and the ribosome in forming a protein. It carries the instructions for building a polypeptide from the gene in DNA to the ribosome in the cytoplasm. So it truly is a messenger. Ribosomal RNA is a structural component of ribosomes, which are primarily made up of rRNA and protein. And along with tRNA, the ribosome helps translate the message of mRNA into a protein. Transfer RNA is actually used to bring the right amino acids to the ribosome. So one side of a transfer RNA is bound to amino acids. The other side is an anticodon that matches the codons of mRNA. It all meets at the ribosome to begin the process of protein synthesis. So messenger RNA truly is our messenger. Just like all RNA, it's single-stranded. We're going to turn the triplet code of DNA into the codon code of mRNA. And the process of turning DNA into RNA is called transcription. At the ribosome, we're going to have rRNA. It combines with proteins to create a 2-subunit organelle that's main goal is protein synthesis. We're going to use the mRNA we made in transcription, the ribosomal RNA, and the transfer RNA to make a protein. The process of turning mRNA into a protein is called translation. So transfer RNA, as the carrier of an amino acid, is going to bring the amino acids we need to link together to make a protein. Once we do finally make a protein again from that mRNA triplet codon that we made, it's going to be considered translation. So protein synthesis you're seeing occurs in 2 steps, transcription and translation. Transcription is called transcription because it's turning DNA into RNA. And DNA and RNA are both nucleic acids. They're really similar. If you transcribe something, you generally transcribe it or take notes in the same language. So DNA and RNA being the same molecular language makes sense that we're going to call that transference or that transition there transcription. Translation is when you change the language. And we're going to change the molecular language by changing a nucleic acid into a protein or polypeptide. So the process at the ribosome where we read mRNA and then decode it and make a polypeptide, that's called translation because we're changing the molecular language. Transcription occurs in the nucleus, whereas translation occurs in the cytoplasm at the ribosome. So you can see here that DNA can be turned into mRNA, which splices out the introns that don't code for a protein. That mRNA transcript then leaves through a nuclear pore and it travels to a ribosome. The ribosome is in red and it gets read through the ribosome, or you can say that the mRNA in blue is going to be threaded through the ribosome, and as the ribosome reads the mRNA, tRNA will bring the appropriate amino acids to build a polypeptide. So transcription is turning DNA into RNA. Transcription specifically makes mRNA. We turn the DNA triplets into mRNA codons. In order to do this, we have to loosen the histones around DNA, basically kind of pull DNA apart so we can read just one side of a DNA strand or template, one side of a gene, or one segment of DNA. RNA polymerase is going to be involved in transcription. Just like DNA polymerase made new DNA strands, RNA polymerase is going to make our mRNA strand that will eventually leave the nucleus. There are 3 processes of transcription: initiation, elongation, and termination. Initiation is the beginning of creating the RNA transcript or creating mRNA. So this involves RNA polymerase separating the DNA strands. Elongation is a process where RNA polymerase keeps adding complementary nucleotides to build mRNA to make that transcript longer. And then finally, transcription will stop when RNA polymerase reaches a termination or stop signal, and that's going to stop the creation of mRNA. And mRNA will detach from the RNA polymerase and move away from the DNA and eventually exit the nucleus once being processed. So again, initiation begins when RNA polymerase binds to a promoter region and begins unzipping DNA and reading the template strand to create the mRNA transcript. The mRNA transcript will get longer and longer during the process of elongation, thanks to RNA polymerase moving along the template strand and joining RNA nucleotides together that are complementary to DNA, specifically the template strand that it's reading. We will rewind the DNA behind the RNA polymerase, and when the RNA polymerase reaches a stop signal, it will come off of the DNA, and the completed mRNA segment will then go to be processed before it leaves the nucleus. The processing of mRNA involves the use of a spliceosome. These are special proteins that remove non-coding regions of mRNA called introns, and they're only going to leave the exon coding regions in the mRNA sequence. Once mRNA has been sequenced and spliced, it then is able to leave the nucleus and it goes to the ribosome where it's going to help convert the base sequence of nucleic acids that we have in RNA into the amino acid sequence of proteins. That process occurs at the ribosome in the cytoplasm and is called translation. Again, the mRNA is going to the ribosome, but we're going to be using tRNA to bring the amino acids, and of course rRNA of the ribosome to help us translate or create our polypeptide. So translation involves mRNA, which means instructions from the genetic code, tRNA to bring the amino acids at the ribosome, and these translating events of, again, initiation, elongation, and termination, and sometimes some rough ER, or rough endoplasmic reticulum. This is a step of protein synthesis where the language of nucleic acids, basically a base sequence, is translated into the language of proteins, which is an amino acid sequence. The genetic code can actually give us an idea of what amino acid is being produced from a DNA triplet. An mRNA codon matches a DNA triplet, and there's 64 possible codons. The 4 bases A, U, C, and G have 3 different places to go, so 4 to the 3rd power is 64. And there are also 3 stop codons, but the rest are codons for amino acids. So there's only really possibly 20 possible amino acids that could come from the DNA triplet code that gets transcribed into mRNA and then translated at the ribosome. There's some redundancy, therefore, to the code. So, for example, some triplets that get turned into codons all code for the same amino acid. So here is a codon chart. Each 3 base sequence on DNA is represented by a codon in mRNA. So a codon is on mRNA. It's the complementary 3 base sequence of mRNA to a DNA triplet. Notice that some amino acids are represented by more than one codon. For example, THR or threonine is represented by ACU, ACC, ACA, and ACG. The way you would read this chart is to find the first base in this column, second base in the top row, and then the third base in the far right column. For example, if I'm trying to find the amino acid sequence or the amino acid that is coded for by the bases A, U, G, I would find A right here, and so that's going to put me somewhere in this row. So I'm going to look at the top. U is this first block, so I'm going to be in this row, now specifically in this block. And if I go to the right, I'm looking for G as my third base, so that's the lower row of that block. So I'm in the first column, first block, last row for AUG. And that's going to code for methionine, or what we consider the start codon. Methionine is the start codon for all amino acid sequences, and that's because it's what's recognized at the ribosome. So after the protein is made, sometimes it is also processed to remove methionine so that all proteins don't actually begin with methionine. They just do right after translation. Then they're processed, folded, and turned into the polypeptide that we need to perform an action or catalyze a reaction. Let's take a better look at tRNA. So on one end of tRNA is an amino acid, and that amino acid is going to match the codon that this anticodon on the other side of tRNA binds to. For example, a tRNA with an anticodon UAU will only be able to load a methionine amino acid to its stem region, because anticodons are at the other end or the opposite head to the amino acid side of a tRNA molecule. So a tRNA is meant to bring amino acid to the ribosome, but they must match something on the mRNA, and they're matching the codons with their own kind of opposite or complementary piece of RNA called the anticodon. The anticodon of tRNA will bind only to the codon on mRNA that is complementary to it. For example, if you have the codon AUA, only the tRNA with the anticodon UAU will match that. So we're still going to see that complementarity that we saw with the 2 DNA strands and then the creation of mRNA. We're going to see ribosomes help coordinate the coupling of mRNA with the anticodons on tRNA. Ribosomes have 2 subunits that come together to read the mRNA and make sure that tRNA is binding in the right order. There are 3 sites on the ribosome. One is the amino acyl site, one is the peptidyl site, and one is the exit site. I like to think of the amino acyl site as the arrival site, the peptidyl site as the processing site, and the exit site as the exit or outgoing location for tRNA. So the tRNA actually arrives at the amino acyl site, moves through and links to the proper mRNA codon at the peptidyl site. The peptidyl site is also going to help us process or create peptide chains or peptide bonds between adjacent amino acids. And then the tRNA, once it's done its job, exits out the exit site. If we're looking at translation, we also have the same three processes that we saw in transcription. The sequence of events in translation are initiation, elongation, and termination. Translation occurs in 3 phases, and those 3 phases require ATP, so this is an active process. Initiation also requires a start codon or a start point, and that's also going to rely on an anticodon to bring the right amino acid, which in this case will be methionine. The elongation involves 3 steps. We need to recognize the first codon, make a peptide bond form multiple times, and then translocate or move that tRNA and mRNA kind of to the next location to make sure that we are constantly processing the mRNA completely and it's not stagnating or getting stuck in the ribosome. Elongation will make sure we're creating a longer amino acid chain. So elongation is referring to the elongation of the polypeptide. Termination is when the ribosome hits one of the 3 stop codons. When the ribosome hits UGA, UAA, or UAG on mRNA, that's going to cause translation to end. Basically, all of these components, the tRNA, mRNA, and ribosome, kind of dissociate or pull apart, and that's going to cause the release of the newly made protein or polypeptide chain, and that will then go off to be processed. So we need to process polypeptide products to make sure their structures turn into their functional 3D protein shapes. So again, translation relies on initiation, so the part where we first start to read mRNA at the ribosome, and tRNA brings the anticodon that is on the other side attached to methionine, so that's the start codon, AUG, that binds to the anticodon, UAC, that is on tRNA bringing methionine. We're going to keep doing that process of bringing tRNA that has amino acids, and they're going to move through the AP then E site. And as we bring more amino acids, they get bound together using peptide bonds. So this process of elongation creates a longer polypeptide. Eventually, a release factor will bind to a stop codon. This actually allows water to bind at the next site, and this water causes a hydrolysis reaction that allows the polypeptide to leave or dissociate from the ribosome. Then everything basically falls apart, and the 2 subunits of a ribosome, the big and the small one, separate as well. So looking at a translation overview, what is happening? Well, translation is the process in which genetic information carried by an mRNA molecule is decoded in the ribosome to form a particular polypeptide. So translation basically is the process that turns mRNA into a protein. It has 3 steps: initiation, elongation, and termination. We rely on the ribosome, tRNA, mRNA, and the creation of a polypeptide thanks to the tRNA holding amino acids on one side and an anticodon on the other. The new amino acids that are added together must be linked with peptide bonds in order to form that elongated polypeptide. And these proteins created have to be further processed. So what we've been looking at is essentially a summary of how DNA gets turned into proteins. And that idea, again, of reading a gene and using that information eventually to make a protein is called the central dogma of molecular biology. It's this idea of using DNA triplets to generate mRNA codons, and then having that mRNA transcript made up of codons leaving the nucleus and going to the cytoplasm and associating with the ribosome and the tRNA and the proper amino acids and essentially making a polypeptide. So the idea of DNA to RNA to proteins, this pathway is the central dogma of molecular biology. Turning DNA into RNA is transcription, and turning RNA into proteins is called translation. So here it is, the central dogma of molecular biology. So if you were asked to write that, you would write DNA, right arrow, RNA, right arrow, protein. So it's a general idea of DNA being the molecular instructions, RNA being the intermediary molecule, and protein being the final product of the process that occurs at the ribosome. This first arrow represents the process of transcription. The second arrow represents the process of translation. And here is just the general process from beginning to end. Notice that on DNA, a group of 3 bases is called a triplet. On mRNA, a group of 3 bases is called a codon. On tRNA, a group of 3 bases is called an anticodon, and the anticodon is on one side of tRNA. On the other side is an amino acid. So we can also see that if this strand of DNA is the template, the mRNA sequence made from it should be complementary, meaning all the bases should pair up like we would expect in 2 opposite DNA strands. So thymine pairs with adenine. Adenine usually pairs with thymine, but remember in RNA we don't have that. It gets replaced by uracil. That's when we see adenine turning into uracil. Cytosine always gets paired with guanine, and we're going to see that repeat. We can create complementary mRNA transcripts by reading a template DNA strand. Now, if we look at a codon, here we have AUG, the anticodon is also complementary. A pairs with U, U pairs with A, G pairs with C. And that's why they match up or recognize each other, and that's how the proper amino acid gets sent to the right codon. That's when we can read a codon chart to figure out what amino acid sequence is being made. All right, our next topic is going to be cell division. So we'll review chromatin and chromosomes and look at how are we going to move this genetic material around. When we're not replicating or reading it, what do we do with it? For cell division. So make sure to read up on that in your textbook so that some of the terminology, a lot of it that begins with C's, is a little bit more recognizable to you.